Tilia platyphilus, and their hybrid, the common or European lime, which is Tilia europaea. In the UK, the small-leaved lime is native to much of England and Wales, but not Scotland, whereas the large-leaved lime is generally less common and has a much more restricted distribution than the small-leaved lime. The European or the common lime has a scattered natural distribution, but it's this tree that we find planted in urban areas, parks, and avenues. In Europe, it's a similar story with the small and large-leaved lime, native to much of Europe, with the small-leaved lime being generally more abundant in Central and Eastern Europe, while the larger-leaved lime has a smaller natural range. There are also two lesser-known species, which are silver lime, Tilia tomentosa, which is commonly found in Hungary and the Balkans, and the Caucasian lime, Tilia dasystyla, which is native to the regions around the Black Sea. Over about the last 2,000 years, the distribution of lime trees in woodlands has declined. However, they are not regarded as threatened in the UK. Tilia provides several ecosystem services. They improve air quality by removing pollution, they store and sequester carbon, and they also support a wide range of biodiversity. However, they are not seen as being valuable in terms of their timber. Now, lime trees do have a reputation of being resilient towards diseases. But despite this, there have been increasing reports of Phytophthora infections in lime trees over the past decade or so. I'm sure you all have heard of Phytophthora. It's a genus that consists of a diverse range of pathogenic oomycetes that are spread through water and soil. And it's a genus that has more than 200 species, causing disease on a range of hosts. The more commonly known ones are probably Phytophthora ramorum and Phytophthora infestans. In lime trees, the cankers on the trunk are usually a common symptom of an infection with Phytophthora plurivera. The cankers stand out as these dark necrotic lesions. These can be either dry or wet, and there's often an exudate that seeps down the trunk. And in extreme cases, bleeding canker can cause dieback of the canopy and even tree death. And there are no effective treatment options. And when several trees are infected, this can have a significant impact on the site. So I'm just going to switch now to a different tree species, and I'm going to tell you just a little bit about acute oak decline, because as you'll see, this does link up to the Tilia project. So acute oak decline, or AOD, is a decline disease. It affects our native oak species, and this is usually on mature trees greater than 50 years old. Symptoms include stem bleeds between the bark plates. This usually occurs in spring and autumn. And when the outer bark is removed, these dark necrotic lesions are present in the underlying tissue. Following repeated infection, it can cause tree death within 3 to 5 years. AOD is caused by both biotic and abiotic factors. There are two bacterial species, Bruneria goodwinii and Gibsiella quercinecans, that are responsible for the tissue necrosis in lesions. And the second biotic factor is the two-spotted oak borer, Agrillus bigotatus, and the larvae of the TSOB is almost always present in close proximity to the lesions, and you can see the feeding galleries here in this image. The abiotic factors of AOD include environmental stresses, such as drought and poor soil condition. So going back to Tilia now, in about 2020, I was working on a large AOD project where one of our objectives was to look for alternate hosts for the bacteria associated with AOD. And lime was actually one of the broadleaf hosts that we wanted to sample to see if we could find the AOD bacteria. So I'd made contacts at Western Arboretum by that stage, and I asked if they just happened to have any Tilia with bleeds, and the manager got back to me and said, yes, they did have a tree with bleeds, and we arranged to go and sample it. But a week before we were due to sample, he contacted me to say that the tree had blown over in a storm. You can see here it was completely over with its roots exposed, which was very sad for the tree, but it did mean that they allowed us to do destructive sampling because we thought we'd only be able to take swabs at that point. So this is the very first bleed that we actually sampled from that tree. You can see it's about, say, 10 centimeters in length at the most. But when we removed the outer bark, we were quite surprised to see that the lesion actually went on a lot further. In this image here, it's about 15 centimeters. And we went on to remove section of the outer bark. And when we finally uncovered the lesion in its entirety, it was close to 1 meter in length. We didn't have anything to measure it with because it was so much longer than we were expecting. And it was samples from this tree that formed the basis of Helena's master's project, where she isolated and identified bacteria from these bleeding cankers. And the results from that master's project revealed that we'd actually isolated a novel Brunneria species, which we formally described as Brunneria tiliae, but we also found and isolated strains of the bacteria associated with acute oak decline, Brunneria goodwinii and Gibsiella quercinecans. Brunneria is a bacterial genus of woody pathogens that are known to cause disease on several different trees, like walnut, willow, and alder. So isolating a new Brunneria species within this genus suggested that it did have some kind of pathogenic potential and could play a role in lesion formation, possibly along with the AOD bacteria. This raised a lot of questions regarding its distribution as well as its role. I was fortunate that I received funding from the Friends of Western Burt Arboretum and UWI, which we could put towards a new project, a PhD, which Helena applied for, and she started this project in January 2023. So the main aims of Helena's PhD are to investigate the microbiome of Tilia suffering from Phytophthora-induced cankers. And this is kind of a two-pronged approach using culturing and identification, as well as metagenomics. We would then compare the microbiomes of diseased and healthy trees and perform pathogenicity tests with potential pathogens. But before we could start with that very first aim of the project, we needed to find trees to sample. We had 3 sites from Helena's master's project, but we needed many more. We did have some historical records of sites from some colleagues of mine at Forest Research, but when we followed up on them, many of them were dead ends and the trees weren't there anymore. But then Helena came up with a brilliant idea of creating an information pamphlet that we could share, asking for people to contact us with reports of bleeding canker on Tilia. And we shared this pamphlet through tree health meetings. We shared it on social media. We asked Forest Research and the Forestry Commission to distribute it for us. And it was also posted on the Bacterial Tree Disease Program website. And we emailed it to a lot of national trust sites as well. And we were absolutely overwhelmed by the response that we've had. We've had local tree wardens, park rangers, arborists, citizen scientists, members of the public emailing us, reporting trees, even offering to send swabs, which has been absolutely fantastic. So the very first year of the project was focused on sampling. Once we had located sites and received permission to visit them and to sample, we went on several field trips and did surveying and sampling of the trees, where we would describe the tree, its general health, its age, and describe the symptoms we were seeing as well of the cankers. We would then take a swab. Sometimes we were allowed to take a small panel, and we would perform Phytophthora LFD tests. So what we noticed as we were going on these field trips was that we were seeing a wide range of symptoms. Sometimes the cankers were very dry, sometimes they were very wet. They varied in length and size. And in the cases where we were allowed to remove a bark panel, sometimes the lesion was very dark with clear margins. Other times it was just a bit of a water-soaked mass on the inner bark. So this is the swab processing procedure that we followed. if we had a swab, usually taken from the runny exudate of the canker, we'd take this back to the lab and spread it on a solid growth media, which would then be incubated for 1 to 2 days. And from this, we would get a range of different bacteria growing, which we then separated out until they were pure. So there was only one kind of bacteria on the plate. The DNA was isolated from this, and then we performed gene sequencing, which allowed us to compare gene that is commonly used for bacterial identification to a whole database of sequences, so we could identify what that pure bacteria was. So this is just seeing that process again. This is a plate of mixed bacterial colonies. You can see the different morphologies there. We'd get a short sequence back. This is maybe 700 base pairs, and it would be compared to a database, and we would get something like this coming up, which says here, Brunaria species. And these are actually ones that we submitted, and this is Brunaria tiliae. If the percentage identification wasn't that high, we can do a comparison where we put it in what we call a phylogenetic tree with other species of Brunaria, and if the sequences fall within certain clusters, we can definitively say the identity of this strain is Brunaria tiliae or not. So just some results from the survey. This is, I think, a map from last year. So at that point, we had more than 60 sites included. I do know this number is higher because we're still receiving reports this year, which is lovely. I'm taking note of these, and Helena will plot them later. Our northernmost reports of bleeding cankers was up in East Lothian in Scotland, and the southernmost was in Plymouth, Devon. The most further east that we've had a report has been in Suffolk, and the most westerly has been Attingham Park in Shrewsbury. But overall, our survey results did vary quite a lot. And what we quickly came to realize is that the general health of the tree didn't always correlate with the severity of the cankers. So you can see here, this is at Attingham Park. This is one of the trees that has a canker, and you can see it's got a lovely, healthy and full crown. But if you look at the cankers on its trunk, they're quite large, they're quite severe, and they're moving up the tree as well. If you compare it now to this site here, this is in Matlock in Derbyshire. The crown of this tree was terrible. It's got severe dieback, and the cankers were very dry and almost scorched. We actually found out early this year from the tree warden who contacted me about this site that this small avenue of trees are actually going to be removed because they've started to pose a safety threat to people walking along the path. And if we take a look now at the results of the bacterial identification and also the Phytophthora results, we identified Phytophthora at every site we tested, which we kind of expected. We frequently isolated Brunaria tiliae as well as the AOD pathogens, although not always together. We isolated Brunaria tiliae from 9 out of 21 sites that we visited, and Brunaria goodwinii and Gibsiella querceneckens were identified from 12 of these 21 sites. So the bacterial identification wasn't really consistent between sites, but this could be due to a range of factors. It could be the different stages of lesion formation that we just happened to sample at, and it was clear that some lesions had progressed further than others. So at the time we sampled, we might have just missed the key bacteria that are in that lesion. The focus of the second year of the project, which was last year, was to compare the microbiome of the lesions. This is the sequencing and identification of all microorganisms in the sample, and this is different to the approach from the first year because there is no culturing step. And this means we can identify microorganisms that we might have missed, but their DNA would still be in the lesion. We've just been unable to culture them. Or that we actually can't culture in the lab because it's just not possible to culture them. We selected 5 sites that had at least 3 symptomatic Tilia on site, and we tried to have a wide distribution throughout the country. So we used a site in Surrey, one in Gloucestershire, one in Derbyshire. We came up to Tatton Park, Cheshire, which is where we met Bronte, and Westerbury Arboretum in Gloucestershire. And where possible as well, we tried to sample from 3 points of the lesion. So typically the top of the lesion, the middle, and the bottom, if we could find it when we took the panel. And we also made sure that we sampled from asymptomatic tissue of all trees as well. So just to take you through the procedure we followed here for this metagenomics, we took sections of bark using a sterilized chisel and hammer. We took these back to the lab and dried them out. And then Helena ground the bark using a handheld Dremel tool. And this was ground into a very fine powder. And from that powder, we isolated the DNA of all microorganisms that would be present. And this was then sent off for whole genome sequencing, which we call metagenomics. And when the genomes are sequenced, it provides us with a lot more information that we can use for identification. The results from the metagenomics came back, I think, 2 weeks before Helena went on maternity leave. So this is very preliminary data that I'm presenting here. But we can already, from this very rough analysis, see that there are differences in the relative abundance of microorganisms in these lesions. If you look at the key for these charts on the side here, the yellow is Brunaria, which is the genus that houses Brunaria tiliae as well as Brunaria goodwinii. And the orange is Pseudomonas, which is also a genus that has some plant pathogens, also some endophytes. And we can see differences in the relative abundance of microbes within the same lesion. So if we look at, this is high Leybourne, the first 9 bars here, and the first 3 bars here are the same lesion, but the bottom, the middle, and the top. And you can see differences in the relative abundance of Brunaria and also differences in the other microorganisms that are found in that lesion. We can also see differences between the microbiomes of trees and lesions between trees at the same site. If we look here at Tatton Park, visually you can see between tree 1, 2, and 3 that the composition of the microbiome, that lesion microbiome, is different between those 3 trees. And finally, we can see differences across the range of sites as well of that lesion microbiome. But we would expect this because we expect that environmental factors would contribute to the microbiome structure of the lesions, and these 5 sites are quite different in terms of some were out in the country, some were a lot more compacted, some were along a path. There were various different environmental reasons for this. But we do need to do a lot more of a deeper analysis on this data. So I mentioned that we took healthy tissue from each tree as well, and we tried to take that tissue on the opposite side of the trunk to the lesion. So in the key here, you can see, if you're eagle-eyed, that the names of the microorganisms here have changed. So the colors that the program uses are the same, but the yellow doesn't represent Brunaria anymore. So what's missing here are the pathogenic bacteria and that we were seeing in the lesion microbiome. And what we can see at a first glance here is that the relative abundance of microbes in the healthy Lyme tissue appears to be a lot more stable, both between trees at the same site and also between the different sites, which in itself is quite interesting. But again, this is something that needs a lot more analysis and also comparing to the diseased data as well. So what do our results mean so far? Well, Phytophthora is likely the main pathogen causing the cankers in Tilia, which we already knew, but there are an abundance of potential bacterial pathogens in those lesion microbiomes as well. And this could indicate that they contribute to disease progression along with the Phytophthora. So there are kind of two ways that the bacteria could be doing this. They could be taking advantage of the situation where the Phytophthora is breaking down the bark tissue and it's releasing an excess of nutrients, which the bacteria loves. This provides an environment for the bacteria, which could be present in the tree in low numbers anyway, to kind of proliferate and come up in greater numbers. Or the bacteria could be secondary invaders from the environment who are taking advantage of a reduced host immunity. And we need a lot more work done on the metagenomic analysis, but our initial analysis indicates that the tree lesion microbiome is significantly different to healthy tilia tissue. So the way forward for this research when Helena returns next year is to do the metagenomic analysis. She will have to do lots and lots of bioinformatic analysis of data sets and do lots of comparisons as well between the lesions, between the trees and the sites, and between the healthy tissue and also a healthy site. We're then going to do pathogenicity tests. We will include selected bacterial strains as well as the Phytophthora oomycete, and we are going to attempt to do this on different Tilia species. We will monitor and measure the lesions that develop through inoculation of these trees and do some statistical analysis to determine if there's any significance. And the expected outcomes from this research will provide understanding of the role of these potential pathogens as well as the threat they may pose. And it will also lead to increased knowledge about Phytophthora infection in Tilia. And what we hope as the final end goal of the project is that we may be able to inform future planting strategies through our pathogenicity test results. So that's it from me, but I would just like to acknowledge the many arborists, tree wardens, Forestry Commission employees, and citizen scientists who sent us swabs or reported trees. We are very grateful for your input. And I'd also like to thank the National Trust sites who allowed us to visit and sample. These were Attingham Park, Clumber Park, Durham Park, Tatton Park, and Tintsfield. And finally, Westernburt Arboretum. And of course, I have to acknowledge our funders, who are the Friends of Westernburt Arboretum and UWE. And thank you for listening, and I'm happy to take questions.
Thank you, Carrie. That was great. I've had a quick peek at the chat, and I haven't got anything in there. So is there anyone who has any questions for Carrie? You're welcome to stick your hand up. A couple more moments for any hands. Well, in which case, I do have a question for you, Carrie. Yeah. You were talking about the microbiome composition. I think that was the metagenomics.
Yes.
And that sort of really broad variation of what you were finding, even on the same site between different samples you'd taken. Is that in part because of the age of the lesion? So perhaps you haven't got as much stuff there. Could that be?
Yeah, I think so. And I think if you can remember what one of the sites looked like, the bars were really quite short, and that was the site where the trees were going to be removed. So that was in Derbyshire. And I think those lesions were just so old by the time we'd got to visit them that nothing was really growing. There was a few things there, but it wasn't a rapidly expanding lesion, put it that way.
Yeah, they're very interesting. I think we've got a hand up from John.
Yep.
If you want to unmute and go ahead with your question.
Yes, there's the AOD bacteria are associated with these lesions. Is the Phytophthora associated with AOD? I've not heard that it is.
No, it's not. That's something that is routinely tested because there's always that concern, but it's not. Acute oak decline is the bacteria, the beetle, and the environmental stresses with no
Okay.
Phytophthora. Yeah.
Thank you.
We've got a question in the chat from Heather Swift. It's a pretty good one, talking about cankers. And you mentioned that they could be wet or dry. Do all wet cankers become dry, and is that at an older stage?
I think possibly, and I would say just rainfall has a lot to do with it and how wet it is. So when we had a very dry period last year, we were worried that we wouldn't find many wet bleeds. But what we have found is often, and we found this at Tatton Park, Bronte, is that the canker appeared so dry, and when Helena put the chisel in and hammered it in, the liquid just shot out, which yeah, missed me by an inch. So that was quite misleading. But
Yeah.
I think also depending on the severity of the canker and how diseased the tree is, it would be a lot drier.
Mm-hmm. And we've got a hand up from Michael as well, and I think we'll take one more question after Michael. If you want to go ahead, Michael.
Yeah, it's not so much a question, it was just an observation. We had an avenue of lime, and we, strangely enough, it was the sort of two trees at either end of the avenue that were struck with the disease. Anyway, a bit of a strange story, but we felled them and planted the stem of one of them to do a chainsaw carving. Possibly it might have been further up than where the lesions were. Anyway, the damn thing started to grow and still growing to this day without any sign of any bleeding lesions on it. So whether the sort of the areas of attack are quite sort of localized and aren't sort of systemic through the whole plant or not, I don't know. But
that's incredible and really interesting. Yeah, that'll definitely give us something to think about. Yeah, thank you.
No problem.
Yeah, I see there's a question in the chat, Bronte, but I can type and reply to that.
Okay, fantastic.
If you want to move on.
Yeah, I've just got one more question actually from myself. Is there anything you want from us and the stakeholders? Are you interested in further reports on bleeds on lime?
Yes, we love the reports to keep coming. The sampling part of the project has to stop, but if there's something out there that might be of a bit more interest, then we could potentially include it in the study. I had someone contacted me a couple of weeks ago saying they had a report on Tilia tomentosa, which we haven't had a sample of yet. So I'm hoping to visit that site.
Okay, yeah, fantastic. And what would be the best way? Is that just emailing you directly?
Email. Yeah, that's great.
Okay, brilliant. Okay. Thank you. In which case we will swiftly move on. Anna, if you can pop your presentation up. So yeah, we've got Anna Platoni with us, who's the Entomology Advisory and Monitoring Manager for Forest Research. And she's got a very interesting talk on plain lacebug.
What's that? Can everyone see that?
I've got that on my screen.
Slides.
Looks good.
Brilliant. Okay, well, thank you for inviting me to talk here today. Like Bronte said, my name is Anna Platoni. I'm the Entomology Advisory and Monitoring Manager for Forest Research. I'm based at Alice Holm. And I've been asked to give you a bit of an update on a pest that was recently discovered in the UK. Or rediscovered, we'll come on to the details of it, called the platinus lacebug. So here's a picture of the beastie. It's from the family Tingidae, which are the lacebugs, and that's in the order Hemiptera. So Hemiptera include also aphids and scale insects, so insects with sucking mouth parts, basically. And you can see why they're called lacebugs. I think they've got these beautiful lacy wings, and there are approximately 2,000 species of lacebugs worldwide. So this particular one, the platinus lacebug, is native to North America, and it feeds on trees from the genus Platanus. So in the US, Platanus is known as sycamore, and this can lead to some confusion in the UK because we use sycamore to refer to Acer pseudoplatanus. And so for this reason, this pest is known as two different names, two different common names. So in the US, you might see it referred to as sycamore lacebug, and in the UK, it's often referred to as plain lacebug. So this is why we're using platinus lacebug, because that's the most accurate and safest common name, though you will see, you know, in I'll say it probably in this presentation, I'll say plain lacebug, and in some of our stuff we're putting out, the information we're putting out is sometimes referred to as plain lacebug, but that's why. And to be safe and clear for the rest of this presentation, I'll probably just be calling it PLB. So in the US, plain is a valued timber tree, and so this pest can be considered a forestry, a pest in a forestry context, and heavy infestations sometimes require chemical treatment, which is, you know, pretty interesting because, you know, in the US we'd expect the highest level of pest suppression from natural enemies that have co-evolved in the same environment as plain lacebug. So the plain lacebug, I've said that it has sucking mouth parts. It feeds on the undersides of the leaves. So you just previously saw a picture of the adult. They're very small, measuring around sort of 4 millimeters in length. The nymphs are black and spiny. You can see the photo here. And they pass through 5 instars, so that's 5 molts, 5 sort of growth stages before they reach maturity. Hemiptera don't do the same thing that, say, beetles or butterflies do, where they go from caterpillar, pupae, adult. These go through these nymphal stages, so basically with each molt, they look more and more like the adult that they will become. One of the other interesting things about PLB is that it overwinters as adults under the bark platelets. So, you know, plain has a very characteristic sort of piece of bark that can flake off. And plain lacebug, PLB, sort of makes the most of this, and it's a good place for it to hide. And the main thing that this does is it makes it extremely tolerant to cold weather. So individuals have been recorded surviving temperatures of up to -30 degrees on the continent. At the other extreme, they've been recorded withstanding hot summer conditions of up to 40 degrees. So despite their delicate appearance, they're pretty hardy things. The season, each female can lay hundreds of eggs, and this means that populations can grow quickly. So the distribution of this pest, PLB was first discovered in Europe. So I've said it's native to Americas. It was first discovered in Europe in 1964, and since then it's spread across much of Central and Southern Europe. More recently, it has spread across parts of Asia, so into Japan and into Australia. Despite the lacy wing coverings, you know, again, that delicate look that it has, it's regarded as a pretty capable flyer. And that's especially when it's wind assisted. And the other thing is it's a known hitchhiker, and we'll come on to that in a bit more detail. But as a hitchhiker, it can be moved from place to place, quite literally off the back of human activity. So the symptoms of this pest. These include, so we've already seen the black nymphs, and the adults on the underside of the leaves, particularly along the veins of the leaves at first, and then sometimes spreading out. And then, the adults on the leaves, the underside of the leaves again, but also, as we mentioned, sheltering under those bark flakes. So quite an important sort of diagnostic feature of this pest is that the nymphs produce black frost droplets, which stick onto the underside of the leaves. And there are lookalikes to this pest. So there are some leafhoppers that will cause similar looking damage to what we'll talk about, but none of them produce these black frost droplets. So if anyone's ever had thrips on their houseplants at home, they produce very similar looking black frost spots. And one of the most useful things about this as a diagnostic feature is, aside from it not being produced by the leafhopper lookalikes, it also can stay on the leaves for a long time. Quite persistent, even maybe into the autumn. And of course, once leaves have fallen, that can be a bit easier to survey. So picking up leaves to taking a look to see if there are any of these black frost droplets that have persisted. Again, there are other things that can cause black dots on plain leaves. But these should be, so these particular ones, the frost droplets, you should be able to scrape them off. You know, they're sticky onto the leaf, but they're not part of the leaf. So you should be able to sort of scratch them off with your finger. Another thing that's good to see is the feeding damage. So we talked about the adults and the nymphs feeding on the underside of the leaves, but on the upper side of the leaves, you can see in this top picture, the feeding damages causes leaf chlorosis, so a loss of leaf colour, and that especially, you know, when you step back, can give the leaves a bronze-like appearance. And aside from all of this, if you find a lacebug on a plain tree, on a plain leaf, that is sufficient really for positive diagnosis, because there are no other lacebugs in the UK that are found on plain trees. So I've got a few more pictures here. So you can see the extent of the black frass on that picture on the left, on the underside of the leaves, and along with the presence of nymphs and adults, so they're coexisting on the same leaf. The other photo shows a leaf with a smaller population, and you can really see them coalescing along the leaf veins. And this can be easily seen. So with a good pair of binoculars, you can see these the nymphs and the black frost spots from the ground. But yeah, you can see them coalescing along the leaf veins, and that's because they're sapsuckers and they want to be closest, at least at first, to the juiciest part of the leaf, basically. So what are the impacts of this pest, or potential impacts? So you can see here some trees that are being affected by the pest, and heavy levels of chlorosis can cause premature leaf fall, so browning and then a leaf fall. And if we think about plane trees and their role in an urban environment, they play an important role in regulating temperatures and providing shade in cities. So they've got a lot of amenity value, and heavy infestations of this pest may impact that. Other than that, however, thankfully, the effects of this pest are largely aesthetic. So the browning of the leaves and the early leaf fall. It's possible, though, that repeated heavy infestations year on year might weaken trees and make them more susceptible to other environmental stresses such as drought, which of course for urban trees, you know, they may already be quite stressed. And then the other thing to note is that at very high densities, PLB can fall from the canopy onto people walking or sitting below trees, and there are records of people being bitten by PLB. and there are even there have been studies that have shown that human blood can be among the gut contents of captured insects. So they can bite people, but usually reactions to these bites are minor. Some people are more sensitive to them than others. So in some people it will produce a rash. So what's the story of PLB in the UK? So I said at the beginning that this most recent finding, we're calling it a rediscovery rather than exactly a first report. And the reason for that is because in 2006, a small population of PLB was found in a nursery environment by AFA in Bedfordshire. The finding was made on some young and imported plants, but it had spread locally to some plane trees in the wider environment. At the time in 2006, some spraying was recommended, but since undertaken. But since then, repeated surveys, including one last year for reasons that will become evident in a moment, have found no further evidence of this pest in that area. So we don't know for sure why the pest didn't persist in this environment. Perhaps the population was just small and was like small enough to fail. That perhaps the spraying was effective in eradicating it. It's also possible sort of a completely untested theory, but it might be that because the plane trees that they arrived on were very young, and of course plain bark becomes sort of more flaky as they get a bit older, it could be that there was limited number of places for the PLB to overwinter. So after this, no further records of PLB in the UK have been made until late July 2024, when a report was made on the web recording platform iNaturalist by a member of the public. They were travelling on a bus through South Kensington, and they spotted an interesting looking insect wandering around on their rucksack. They snapped a blurry photo. Now we see quite a lot of blurry photos in different different levels of blurriness, let's say. We consider ourselves sort of blurry photo experts, and this was clear enough to set alarm bells ringing. So a colleague in DEFRA had spotted the iNaturalist report and flagged it with FERA. FR and AFA, we followed up in the field, and although just having this picture to go on, we knew the bus route, so started to follow the bus route to see what we could find, and lo and behold, we found roughly 40 street and parkland trees found to be infested with PLB. So what did we do next? So there was a survey response to this pest. The aim was to find out, was it simply in this one area or further beyond? So it prompted a coordinated survey effort. So used contractors to survey plane trees across London, especially they'd just been surveying plane trees for plain wilt. Unfortunately, it just finished, but they were sent out again to look at the trees. So they were resurveyed. And in addition to this, which was sort of, it's like a designed survey that was pre-existing, FC and FR staff then carried out surveys in highly trafficked areas. So we didn't know how PLB had arrived here. but given, like we mentioned earlier, you know, it's got a proven propensity to hitchhike, we prioritised areas of high footfall, so including bus and train stations. And over the course of the next sort of couple of months, of course, this was sort of, you know, August by the time the surveys were kicking off in earnest, and so we were fighting against the onset of autumn. Over the next couple of months, over 1,600 trees in the genus Platanus were surveyed, and the great majority of these were London plane, mainly because that's what that is the main plane you find in London. And this map shows all of the trees that were surveyed. So as part of this survey effort, 2 further populations of PLB were found of a similar size to that first finding, again in other busy areas of central London. So one in the King's Cross area by Chris Malumphy from Ferrer during a trip through the city, and the other was on Thames Embankment by FR colleagues. So since that surveillance was carried out, so that gave us 3 known populations described in London. A further report has since been made on iNaturalist, and this one is in the Russell Square area of London, and that was of a Platanus lacebug adult overwintering under plain bark. So in addition to the organised surveys, a call was also sent out through the observatory platform to the volunteers to ask them to survey plane in their local areas. To help them do this, we produced an ID supplement guide. So plain lacebug is already a priority pest for observatory volunteers, so they already had some information about it, but a group of us having spent quite a long time surveying ourselves, that personal experience then gives you sort of more insight into the potential pitfalls, the lookalikes, had access to more photos, and yeah, just generally more experience. So we then produced an extra guide, a supplementary guide. And both of these, the original and the supplementary guide, are both available on the observatory website. But yeah, the hope for this supplementary guide was to help the volunteers distinguish between PLB and other common insects and diseases found on plane, which was an attempt to reduce the false positives, and that seems to have worked really well. So this was essentially a call to action for the observatory volunteers, and it was delivered as part of a host of the month webinar that the volunteers attended in the September, and then they had a question and answer session with the scientists for the volunteers to learn more. So control options. It's not a regulated pest, and that means that there's no statutory requirement to take action. We know that there are now, well, 3 centres of infestation that we found with areas with, so it looks like there's strong centres and then areas of lower density found surrounding these areas. And then this other extra sighting found. So given the dispersal capability of this pest, there could well be more further afield, and one of the things I'm going to be asking you guys to do is to keep a lookout on your plane trees and help us answer this question essentially. But given that populations, or those populations that we already know about, eradication is most likely impossible. And then on top of that, spraying using pesticides or a nematode or fungi biocontrol, they have limited efficacy for the pest, especially because the adults can shelter under bark flakes where they're protected. And in addition, I use this picture here to illustrate just to think about what that would mean to use spray or trunk injections on these trees, with such highly trafficked areas. So that would likely come in with public opposition. We also have an example, we can see on the continent that where populations of this pest are often very high, they don't use control methods routinely for the pest. And on top of all of that, the impacts for this pest, as we discussed, are largely aesthetic. And so given all of this, it's been concluded that no action will be taken in this instance, and that landowners and managers will be provided with guidance where it's required. So speaking of that, I've got some places where you can find some more information. So I mentioned the observatory webpage already. We've also got an FR webpage about the biology. We've just finished producing a public information leaflet, which can be seen there on the right. And hopefully I'll be able to circulate that soon, and hopefully it'll go on the websites. We would like FR to do some more research into plain lacebug, because we've got some questions that we'd like to answer in a GB context, most especially how many generations will PLB be able to complete? Our climate can be quite different to some of the places in Europe where it's already found, so that would be particularly interesting, but that will be based on funding. And now, because Bronte very kindly gave me a long slot, I wonder if you indulge me, and we'll look at something, and I say slightly different because this is still a Hemipteran pest. So this is a Holm oak bark scale, which will forever, at least in my mind, be quite linked to the plain lacebug, because we discovered them on the same day in London. So this was, as well as we'd had the iNaturalist report for plain lacebug, we'd also had a tree alert reporting this pest. And so in an attack of efficiency, let's call it, I thought when planning the field work, a team's going into London, let's cover both on the same day. How likely is it that both of these are actually new insects to the UK? Well, the answer was both of them. So yeah, they are linked. It's the same, found in the same summer, and they're both Hemipteran pests. But other than that, as you can see from these pictures, they're pretty different beasts. So rather than a foliar pest, this lives on the bark of Holm oak. So it's a scale insect that's native to the Mediterranean, and its main host is Quercus ilex, but it also can infest other evergreen oaks. And like I said, this was the first discovery of it in the UK in an outbreak in central London, and again in August 2024. So since the finding of this first outbreak via a tree alert, again, we've done further surveys, and we found some populations around the site of that first finding. We don't know how long they've been present. That's probably been quite a few years, and we're still not 100% sure on a pathway how they might have got here. So the symptoms, you can see that this picture of the whole tree, infested trees, especially in this first outbreak site, exhibited canopy decline, some dieback, and also galling on the apical twigs. On the very heavily infested trees, and this wasn't always the case, the scale insects themselves are sort of very clear, almost even from a distance. So on these pictures, you can see the brown dots are the females, and they're sitting on top of the white, their white eggs. So yeah, they're feeding by putting their stylets. So again, they're an insect with sucking mouth parts. So they feed by inserting their stylet mouth parts into the tree, so into the bark, and establishing themselves in bark crevices. So they like the crevices because the bark is a bit softer there. But then once they're there, they can increase the size. They cause the increase of size of the bark crevices. So sort of make their home a bit nicer. We've got a generic sort of schematic of a scale insect life cycle. But out of those tiny eggs that the females are sitting on, the first stage that come out of there is called a crawler, and they're the ones that are mobile. So the adult females is where scale insects get their name from, and that's because they sort of lose their legs, lose their mobility. But the very first stage, which are incredibly small, a couple of millimeters long, or even less, they're the ones that are mobile. They've got legs, and they can also be blown by the wind or via human activity. So again, brushing past or on tools. We're having a look into, it's possible that stress trees will be more susceptible to this pest, and there's a chance that there's some sort of fungi that maybe are associated with the scale insect as well. It's likely that they have one generation a year. Each female will lay 100, 400 eggs in the spring, and then the adult females diapause from the late spring to summer until the following early spring. They're not the only things that can look like, they're quite distinct when you see them, but there are some lookalike things, especially some fungi in bark crevices, especially when you're like sort of standing back, can look very similar. So that's all from me, but I just wanted to shout out a few thanks, mainly to people from FR, FC, AFA, and DEFRA, and also the contractors that worked on the surveys and the observatory volunteers.
Thanks, Anna.
And I'd be really happy to answer any questions. But yeah, the big plea is if you see either of those pests, to drop us a line over on Tree Alert.
Excellent. No, good to plug Tree Alert. Fantastic. I think we've got a bit of time for some questions, and I think we had one pop up whilst you were talking in the chat. So we've got Rick here who's asking, PRB has no natural predators in the UK, so chem control may be necessary. What is it predated by in its natural environments? You've got a couple of questions in there. Go on.
Oh yeah, go on. I was just going to say that we know that like ladybirds and spiders will eat it in its natural environment. So hopefully, we've got related species here. So there is a hope that there will be some biocontrol.
Fantastic. So we do have some natural predators in the UK, and I think, like you said in the talk, there isn't really any benefit from chem control, the situation we're in. Excellent. Also in the chat from Sophie Price, she sort of plugged Pest Watch, which is a project by DEFRA and Forest Research with iNaturalist. From your perspective, I know you've got Tree Alert up there. Is that the preferred option for reporting if people do spot things outside?
Yeah, for us, always Tree Alert. we do monitor iNaturalist, but the advantage of Tree Alert for us is that we get all of the we ask a lot of questions, which I know makes the form, you know, can feel a bit arduous to fill in, but it means we come back to you with a lot fewer questions. It also means we definitely got your contact details and yeah, a lot of the information we need to follow up on things. iNaturalist, although we monitor it, it can then be a bit difficult to you know, find where was meant, contact the person that's reported. So yeah, it's really monitor that just to check in with people that might not know about Tree Alert essentially. But Tree Alert would always be our first preference for you submitting stuff.
Perfect. And if anyone's got any questions, if they want to raise their hands, I will give that a moment. And also I'll say, Anna, that was very well timed. That's what I've got written down on my piece of paper, 11:05.
Perfect, thank you.
If there's no hands going up, I do have a quick question. Sort of thinking about the Northwest, and you're sort of talking about how many generations can be produced in a year. Do you think the climate up here will mean it's less likely to persist if it did come up here?
Yeah, it's hard to know. We don't even know, you know, both of those pests. We've really only got data from countries where the climate is quite different, even from the southeast of England. And of course, London is, you know, an urban heat island, even within that. So my suspicion would be that it would be more difficult for it to establish, and we'll have fewer generations potentially, and especially with plain lacebug, it's the number of generations that will increase the amount of damage. So, you know, saying that, you know, I've said how hardy plain lacebug is, so it doesn't mean that it can't and won't probably. So yeah, would still really appreciate you keeping an eye out, especially in cities where there's likely to be that warmer temperature. One thing to mention on that is that I talked about the surveying by looking under bark flakes. So at one point during last summer, we were considering a campaign called Peel a Plane to get people to survey for them. But what we were finding, especially sort of the back end of August and early in September, that it was only on the very heavily infested trees, so the trees where we could really see a lot of damage on the leaves, where we were reliably finding adults under the bark. And I think that's just probably, you know, a numbers game essentially. There can be a lot of trunk for them to hide under, and unless there's a lot of insects, then when you take a piece of bark off, you won't reliably get one underneath there. So what that does mean is if you have fewer generations, fewer insects in the Northwest, then at that point that might become a less useful survey method. So that's to say that just because, you know, you look under the bark and don't find any, I guess it doesn't necessarily mean it's not there somewhere up in the tree.
Yeah, so we'll be out and about staring upwards at leaves and risking the threat of PLB falling on us and giving us a nasty bite. No, that's great, Anna. If there aren't any other questions, I think we'll move on to Mick. So I think, Anna, if your presentation can go, and then Mick will be able to stick his up. Haven't got the presentation up yet, Mick. We've just got a picture of your Windows background. And now we've got a view of the presenter view, so we've got your notes and stuff as well. I'll just introduce you while you're doing that. So
always helpful, always.
Mick Biddle is a pathology response manager for forest research, and he's been doing an interesting sort of campaign trying to get people involved with this tree health survey, and it's going to be more plain talk. Yeah, it's going to be an interesting one and something hopefully everyone can get involved with.
How's that?
There we go. Yep, got it. Perfect.
Okay, sorry about that. I tried that before the meeting and it worked perfectly, and then for some reason didn't work then. Thanks, Bronte, for that introduction, and thanks for having me here today. And you're quite right, I'm here with an ulterior motive to plug this this survey which we're undertaking at the moment. But it's a kind of clever way of hiding the fact that I'm also going to be talking about plane trees and in this case a pathogen threat to plane trees. I'll start by introducing myself. I'm Mick Biddle. For 3 years now, I've been the pathology response manager for Forest Research, and part of that role is improving preparedness for tree health threats and being able to respond to tree health pathogen threats. Prior to that, I don't have a scientific background particularly. I came via the Forestry Commission plant health forestry team, so I was 12 years working with Bronte, and prior to Bronte, Barnaby, who many of you will know. And I can see I was browsing the attendees list, I can see some really familiar names that I've come across over the years. So hello to everyone, and everyone who doesn't know me as well. So I'm going to talk, I'll come back to the survey at the end, but I'm going to talk about this disease, canker stain of plane, also known as plane wilt. And I mentioned some plane wilt surveys. And again, in America, they call it canker of sycamore. But for the purposes of this, canker stain of plane is the common name that I'm going to use, or CSP. I'll give you a brief overview of the background to the disease, the symptoms, look at the impacts, and then tell you about some of the research that we're doing in preparedness for this tree health threat. So I'll start off by saying, I mean, when I started this role in 2022, one of the things that I wanted to do was improve preparedness and awareness of this disease, because it was one of the ones that was quite a large threat to the urban treescapes. And as part of that, in 2023, I organised a visit with an expert in the south of France, in Avignon. So the guy on the right there is Francis Maire, who deals with this disease in the Provence and the Avignon area. And we had a couple of days with him. So there's myself, Alex Lewis from Forest Research, and then 2 colleagues, Ned and Chloe from Forestry Commission, all went to learn what we could and improve our knowledge about this disease. And it was a really fantastic couple of days and really worthwhile. And off the back of that, then we've had some research projects informed and kicked off. So a lot of the photographs I'm about to show you are from this trip in 2023. The disease itself, canker stain of plane, caused by a fungus, Ceratocystis platani, and it kills plane trees. And as Anna's already mentioned, particularly London plane to the UK, really highly important species to our urban treescapes. It can tolerate pollution really well. It copes with drought, and it has a really positive effect in the cooling. It casts a very cooling shade, which is really important. So these make it an ideal species for an urban location. And anything that threatens the health of the plane trees is something to be concerned about from that point of view. And this disease very much does that. It causes mortality of plane species, particularly London plane and Oriental plane, less so the American Occidentalis. It's spread principally by human activity. It can produce spores. That's pictures of the perithecia on at the bottom right there. But principally it's spread by human activity, and it can travel in water. And then some insects have been shown to be able to spread this disease as well. Although the plain lacebug doesn't appear to spread the disease. So that's some good news. That can cause tree death in this disease can cause death of trees in as little as 2 years, commonly a little bit longer than that for mature trees, but it's a out and out tree killer. And there are no biological or chemical controls available. There's no resistance because many, all of the plane trees, the London plane trees particularly, are clonal, and the resistance, the genetic diversity of them is not great. Therefore, there's no known resistance in sort of mature trees. There is a resistant variety which has been developed, but it's not completely immune. I'll come on to talk about that later. Looking at the threat on the plant health risk register, the unmitigated threat, it scores very highly due to the large impact and the ability of this disease to spread. It's as a consequence, it's a regulated quarantine pest, and there are import requirements on any plane trees or any plane material coming into the UK. It's subject to plant health controls for high risk goods, so any plane trees must have been grown throughout their lifetime in an area that's shown to be free of the pest. And we've had surveys undertaken in the UK since 2014, and we've got a pest-specific contingency plan in place since 2016. And those mitigations have reduced the risk, according to the risk register, down to a more acceptable level. But it still poses a threat, and it's still something we shouldn't be we shouldn't be complacent about. Looking at the distribution of the pathogen, it was native to the eastern USA, and it was introduced to Europe during the Second World War, probably on military movement of ammunition crates and packaging, introduced to Italy and to the south of France, where it's been spreading ever since. Then it spread from Italy into Switzerland, and then also it's been picked up in Greece and spread then to Turkey and also Albania as well. And interestingly, in 2010, there was a finding in Spain, which was associated with movement of machinery from France, but that has been eradicated. So that's good news. Sometime during that period in the 80s, sometime it was introduced from the eastern to the western side of the US as well. Looking at our sort of closest neighbours, France, it's predominantly the south of France that's affected, the Avignon-Provence area. It's also had a huge impact along the Canal du Midi, which is a very famous canal lined completely with plane trees, or it was, but there's been over 100,000 plane trees removed due to this disease along the canal. And the two more northerly outbreaks in Paris and Nantes are thought to be associated. We learned from Francis, he thinks they're associated with the movement of civil engineering road building machinery, which is a pathway that hasn't really been evaluated fully from a UK point of view. So it can move, although relatively, it's been there decades in the south of France. It's moved relatively slowly, but the human assisted movement is the main pathway really. And then talking of which, it can travel in water. It can be moved, or it's a wound pathogen. So any damage to trees alongside waterways where there's infected material can be a point of entry. So particularly along the Canal du Midi, boats banging up against roots or people mooring their boats to trees could be potential points of entry. One of the main ways that it can spread, though, is through arborists' tools. It spreads in sawdust, and plane's a really, really dusty tree when you start working on it, and the sawdust can spread up to 200 meters, or infective sawdust has been found up to 200 meters away from operating on infected trees. And that's the principal way, once it gets established in a country, that it's moved around on pruning, because obviously plane is pollarded very regularly in urban locations. But it can also be spread around anything that causes wounds. Essentially, obviously, if people are planting infected plane trees, then they can be moved around that way. But mowing damage is another way that it can be moved around. And as I said, civil engineering, so earth workers, people who are not necessarily interested or looking at the trees that they're operating in their location, they could be moving it around. It can travel in soil and bits of infected root material as well that could be moved around. Once it gets introduced to the tree, on the left there, that's a pruning wound where infection is initiated. You get this canker starting to develop in the canopy, and eventually that makes its way down the tree to the roots, and then often planes are planted in avenues or in lines, and the root to root contact then allows a pathway along to the next tree along, and it girdles the tree from the base up then. So rather than an individual branch dying, you get the whole tree suddenly wilting, which is why it's got the name plain wilt. So that's the way it can spread kind of naturally. It also seems to be able to reproduce and produce spores on recent wounds where it's been infected. But principally, as I say, it's the human-assisted movement, which is the way it gets around, or where it gets introduced, and then it wakes its way slowly along the lines. And in real life, this is what it can look like. It's a picture on the left from Italy, where it's likely been introduced to that tree on the left during the previous pruning operations at the top right of the tree there. And then that tree on the right hand side of the screen is one that's been girdled or about to be girdled at the base through movement from root to root. In association with the dieback, you get these are the cankers, these subcortical cankers that can look like streaks or leopard spots. You've got on the left here, I don't know if you can see my cursor, but you've got the natural bark plates here, which are these darker areas. But then underneath the bark, you've got this sort of leopard spotting effect and these streaks. That's these cankers that develop on the stem, and they can have dark centers and sort of yellow and purplish margins. These are the healthy bark plates again on the right, but it's this subcortical cankers which you see. Something we learned in France was they can be spotted through the thin bark of plane trees, but it's much easier when you wet the bark. Francis used an alcohol, but it just worked with spraying water on them. It showed up much easier. So that was a really good thing to learn. I think in the UK, the clones that we have over here seem to be thicker bark than those in the south of France and other places in southern Europe. So it might be more difficult to see through the thicker barked planes, but hopefully we won't find out. Where we've got older cankers, you get these longitudinal kind of cracks occurring, and where the cankers start to kill the bark, one of the diagnostic features of the dead bark is that it cracks into kind of square or roughly rectangular kind of plates. You can see on the right here. And then underneath the bark, when you scrape away the cambium, you get staining in the xylem, which extends right into the center of the tree along the medullary rays, and that's what gives this stain, the canker stain, which gives the disease its common name. You see under the bark. So looking at the impacts, it was really shocking. I don't know if many people have been to Avignon, beautiful UNESCO World Heritage walled medieval city, and it's full of plane trees throughout and also around the edges of the walls, and most of the plane trees were infected or in the dying, dead, or in the process of being removed or already gone. So these pictures are taken in late August. So obviously these trees should be in full leaf, but they're not. And this was a kind of tourist honey trap in the centre of Avignon. Beautiful street with lovely boutique shops, and all of the trees were dying alongside this area. And what was really interesting, as well as the obvious visual impacts and the difficulties associated with that, the social impacts of this disease were quite clear as well, because where these monoliths had been left while they were trying to deal with these trees and get them removed, each one of them, someone had gone and written various bits of graffiti. This one means on the left, "Thus dies our culture," and also people had written poems and pinned them to these trees. So they obviously mean an awful lot to the residents of Avignon. And similarly, I think there'll be a similar sort of social, cultural attachment to those in the UK. So really interesting observation. The control measures for the disease are very draconian. There's no other, as I say, there's no other way of dealing with it other than trying to fell the trees and stop the spread that way. Involves the stump removal, and in a place like Avignon, it was hugely difficult. And they're kind of in a cleanup operation there. Really, they're not at the point of being able to stop the spread in that area, because it's so difficult to work on these trees with all the archaeological constraints and the heritage constraints that they're working in. So they kind of monolith the trees, and then eventually will remove the stumps, the high stumps, and you can see more of this graffiti on these trees as well. But these were just notice this same: don't park underneath these trees because they're going to be removed soon. And Francis took us to because the arisings are infective as well, so these have to be dealt with, essentially incinerated to be safely disposed of. But it's a difficult thing to arrange, and the arisings are obviously huge in an area where you've got heavy infestation. So this was a huge kind of holding area where infective arisings were kept before they could be then shipped off for burning in biomass. So pretty bad news, very depressing couple of days. On the plus side, I mentioned this resistant variety which has been developed, which has been given the brand name Platinor, and you can see those. This was the square in the centre of Avignon, and the trees have been removed and replaced by this resistant variety, and they seem to be doing pretty well. And this was further down that street on the right, where you've got all those dead hulks of old trees. Some of the replacement trees were looking quite healthy, but there were, it's not completely immune. So these trees are being planted in the infected planting pits of trees that were heavily infected. So they're under a high pressure, and some of them in this bar area, although they look principally healthy, there were one or two that didn't look healthy, and it has been confirmed as susceptible. So probably not the only answer. It forms a useful replacement species to use in certain circumstances, but of course you're narrowing your genetic base even further by putting all your eggs in this one basket. So if something else comes along, then it could be damaging. Or if the trees get to a certain age and then suddenly decide they are susceptible to the disease, then there's a risk. So this was the parting shot really from Francis. A tip for our English friends: try to stay without canker stain of plain, which kind of stuck with us. And off the back of this visit, we put together some proposals for 3 preparedness projects, which have been funded by the DEFRA Future Proofing Plant Health Scheme. And the first project, which is being led by a colleague, Caroline Gorton, is to improve our diagnostic capability and to establish some level of surveillance using spore trapping rather than visual inspection. So that's this network of spore traps has been set up using mainly botanical gardens across the UK to look for a well, that'll be monitoring for a local source of inoculum. We're not expecting, we're hoping not to find it. We're not expecting to find positives, but it's a useful surveillance technique. We've also had positive material received from France, which we've been able to work on in our quarantine facility, and working with FERA, comparing diagnostic methods and developing digital and real-time PCR methods. And we've also exploring just different sort of culture and baiting techniques that to allow us to be in the best possible position to be able to diagnose things accurately and quickly if we ever had an outbreak in the UK. I don't think I actually said at the start that we haven't had it in the UK. I should have probably opened with that one. It's not something that we found so far in the UK, luckily. The second project is being led by Jen Clements, colleague at Forest Research, looking at the socio-cultural value of plane and looking also at the distribution, and so improving our understanding of the distribution of plane to aid surveillance and aid preparedness in that way, and also inform messaging to increase awareness about this disease. And progress on this so far, a literature review's been completed, which looking at the socio-cultural value of plane, and I mean it's work in progress, this project, but one of the key takeaways from this literature review was that the ecosystem services provided by plane remain understudied. So that's probably a recommendation that's looked at in a bit more detail. There have also been in situ surveys undertaken for public perceptions of plane trees, undertaken in London, York and Bristol. And the majority of people that were spoken to had a positive feeling about plane trees, which is good news. There was some fairly strong views pro and against the pollarding techniques that are used to manage them. And 50% of people could identify the species, which I think is actually quite impressive, given the similarity of the leaf shape to acers. And the other aspect, this distribution mapping, so a machine learning model's been developed using satellite imagery. So the machine learning model's been developed, which is able to use satellite imagery to identify plane trees. So this is an example of the output in London from there. So it's able to predict with quite a high degree of accuracy where plane is from the analysis of satellite images. And we've got verification that we need to complete that this year. It must be said that the accuracy was better in the south of England than it has been in the north of England, but that's because of the amount of plane present in south versus north and the data that we were able to feed into the model to develop it. But there's still scope for it to improve. And then the final one, which is the one that I'm leading, is to do with pathways and practices associated with this disease. So does the movement of machinery and arb equipment present a significant pathway for the disease to enter and spread within the UK? And we're looking at speaking to managers like Francis across other affected countries in Europe, so that we can improve our contingency planning and develop our methodology and plan for an outbreak if it was ever to occur in the UK. And progress on that has been good. There's the project team there. We've undertaken a kind of desk review of the European management practices, and some recommendations from that are starting to emerge. Things like timing of operations to minimise spread, the availability of a toolbox to allow people to develop a management plan. Where if you've got a lot of plane trees, it makes sense to have a plan in place yourself, not just leaving it to the government to work it out for you. But these are some of the recommendations that are starting to come out, but there's more to come from that. One of the places where we've had trouble engaging with is the civil engineering sector. We wanted to undertake interviews with civil engineers, but unfortunately it's been difficult to engage with them, really. So we've got a survey. We're trying to hit as many of people as we can through that this year. And we've also got the interviews with the Cancastena plane experts in the other countries to come later this year as well. And then moving on to the main thrust, which is this arboriculture survey. So this was launched in March in order to gauge awareness of this disease and to have get a better understanding of the movement of arborists backwards and forwards between infected countries and the UK, and also get an understanding of how well biosecurity is observed within the sector as well. So that was launched in March. It's an online survey, and we've got a reasonably successful comms campaign. We've had over 300 respondents so far, and I was at the ARB show pushing it quite hard there and managed to speak to lots of people. So that is the link to the survey. We're particularly after arboriculture professionals for this one. So if you deal with tree surgery or tree officers, anyway, arboricultural professionals, you'll know who you are. I think if we're interested in hearing from you, and it's take about 10 minutes via this QR code. That it can be completed anonymously. You do have the option to leave your email address if you want to hear from us. But yeah, I would urge people please to spread the word and to fill it in where you can. Thank you.
Thanks, Mick. I will also share that link with a follow-up email. So if you've missed that QR code, I'll send that out. So we've got time for some questions. Just looking at the chat, I've got one here from Ruth. She's willing to share that with their contractor, so that's excellent. You've already got some takers.
Brilliant.
Have you got any questions? If people want to raise hands, give that a moment whilst people think. I have a question, Mick. Sort of thinking about the sort of pathways of how it's going to be introduced, or suspected pathways. Could we anticipate it sort of rocking up in the southeast? Not that I have any ill feelings towards the southeast, but you know, would we have some advance warning here in the northwest, I guess?
I mean, it depends on the method of arrival, really. If for example, it arrived on a chainsaw or something that had been used on an infected tree in Italy, and it just depends where that chainsaw is next used. If it comes into contact with a tree, so it could crop up anywhere. And you know, there are regulations around the import of plain trees, so that's probably less of a risk. But if it was introduced on planting, for example, that could come up anywhere. And you know, Manchester, Liverpool, they're full of plains.
Yeah.
So yeah, could crop up anywhere. I mean, the climatic, it grows best at 25 degrees C, but it can tolerate lower temperatures than that. But it's probably at the moment for the UK, it's less. We would be less susceptible than Southern Europe, for example. But as you say, with the heat effect of urban areas and changing climate, that's not to say it isn't a risk. And also, it can survive at lower temperatures. We don't know quite how it would behave in a UK climate.
Yeah. We've got a hand up here from Michael. If you want to go ahead.
Hi, Mick. Yeah, I was just wondering what they were doing with all these trees that they're having to fell over on the continent. You know, are they having to dispose of it in some specific way, given how contagious it is, or are they actually reusing the wood?
No, it's being burned. Essentially, has to be incinerated.
Ouch. That's good, isn't it?
Yeah, yeah. Hence that big pile of arisings, you know, that they're waiting to be disposed of, because they've got so much. And like I say, it's a cleanup operation at the moment in the south of France and large parts of Italy as well. I think in Switzerland there's more of an eradication footing, and the northern areas of France they're under eradication. And we really want to speak to colleagues in Spain that have managed the successful eradication there, and what we can learn from them, as well as speaking to the people that are, you know, dealing with it day in, day out. But yeah, it's one that would be severely damaging if it arrived.
I've got one last question. You spoke about, if I remember the name correctly, Planator. That is it, the variety sort of tolerant to this. Is there anything else on the horizon? You're sort of saying about not putting all our eggs in one basket.
Not that I'm aware of at the moment. That's the only. There was a large number of cultivars and clones that were and crosses that were analysed, and that was what developed from that. But yeah, not at the moment. I mean, it is being planted and it is being used, but it's not completely immune.
Yeah.
So that's the danger with doing that. I mean, the Platanus occidentalis, the American plane, is less susceptible than Orientalis and the London plane.
Oh, brilliant, thank you. Well, yeah, like I said, I'll share the link with everyone after this.
Thank you.
So you can go and share with contractors and things and submit reports. Right,
I'll pop the link to the. I'll pop the link to the FR page in the chat as well. There's a little explainer video as well that I'll pull people towards. But thank you very much.
Thank you. Right, well, I guess then it's now my turn to stick my presentation up on the screen. So if you bear with. Mick, could you tell me if you've just got the slides or if you got all my notes and stuff as well?
Sorry, I was on mute. I think we can see that fine. Yeah.
Yeah,
got the slides.
Perfect. Right. So yes, I'm going to rapidly go through this presentation, looking at green spruce aphid and basically what we've been seeing in the northwest. So I'll be explaining a bit about the pest, but mainly sort of sharing photos and thoughts about what we've been seeing this year. So we've been getting lots of phone calls and emails from concerned landowners and agents about their spruce. And I'm sure many of you, especially up in Cumbria, can recognise this, sort of people talking about all their Sitka and also mentioning Norway spruce being browned off. So I'm mentioning 100 acres of spruce turned ginger, and some people even thinking, you know, all my spruce has suddenly died, sort of coming into late spring. So I had lots of these reports, and all those comments have been reflected in our aerial surveillance. So this is a photo from our flight across the northwest at the end of May, so last month. And you can see that spruce sort of far off in the distance, looking unusually brown. So reflecting those comments that people shared with us. So the culprit, obviously the title gave it away. It's this Elatobium abietinum, this green spruce aphid, which you can see on the screen here, perched on a needle. So it's active from March to May, but it's present all year round. You get peak populations at the end of May, sort of depending on the weather. But then once the needles have flushed, you get the population sort of drops off, dies off, and then you get a further reduction in winter. So if you wandered out now to go have a look at your poorly spruce trees, you're unlikely to spot the aphid, but obviously you're going to see the signs of it having been there with all this defoliation. So again, taking a closer look at the damage that these aphids can cause. They sort of are going in, feeding late winter, early spring, feeding on old needles, getting all the nutrients, because obviously you've got all that resin pressure and all the nutrients building up on those old needles to sort of support the bud development for the new year. So really nutritious stuff. So the aphids are feeding on that. And it's actually the toxins that the aphids secrete as they're feeding that cause this chlorosis. So you get chlorotic spots and banding on these old needles, and then these can coalesce and sort of cause browning of the needles as time goes on. And then subsequently, as we've seen sort of in late spring, coming into May, the needles will drop and then you get this defoliation. But the key thing to point out here is that it's the old needles. So as you can see sort of in this photo here, you've got all that brown old needles, but you can still see that the new sort of buds ready to flush that are still green and healthy. So we know the tree's actually all right. It's just losing those old needles. And this is a closer look at that sort of chlorosis that you could probably see over late winter, sort of that initial feeding on the needles. So just again a bit more about how it works. So typically get a boom year every 3 to 6 years. And then I'm happy to say that typically after a boom year, you then get a drop in the damage, and this is because of a really strong negative feedback loop with aphid populations. So where you get a really big increase in aphid populations, you then get an increase in predators and parasites that obviously feed on them. So then you can expect those to persist into the next year. So we're sort of thinking 2026 into that spring. It then obviously brings the number of aphids down. So you don't get these sort of boom years back to back because you've got those sort of predators and things out there. Also because of the nutrients available is less. So you sort of think all those old needles that the aphid are feeding on this year, it's going to take a few years for those to come back. So you haven't got enough old needles and nutrients to support larger populations the subsequent year. So that's why you get this drop after a boom year. Sort of thinking about what favours the aphid. So mild winters, I'm sure we can all recall the winter just gone, particularly mild, and that favours increased population densities, because obviously you don't get such a drop in the population over winter due to a decrease in temperatures. And I'm also happy to say that there's no long-term impact on the tree's health. So again, thinking about you've still got those green ends to your branches, so you're still going to get that flush of new needles, take a few years for it to sort of recover from the defoliation, but ultimately no long-term impact. So yeah, thinking about this boom year for Northwest, we've got some lovely photos to share. So these are some photos taken from that aerial flight at the end of May. And what we saw generally was a lot more defoliation and stronger symptoms, especially in Cumbria. So we had a flight going through Lancashire, a bit into Yorkshire as well, but the symptoms were most prominent in Cumbria. And I think I spoke to Gwen yesterday, who's our support officer in the Northwest, and she's sort of been out and about driving around, and she said that between, I think there's Winlatter and Penrith on the A66 is a perfect spot to go and see some really, prominent defoliation on the spruce there from the road. So if anyone wants to go out and see it in person who hasn't spotted it yet. But yeah, so this is a photo from between Cockermouth and Penrith, so sort of in that vicinity, and you can sort of see really that sort of gingering that people were mentioning in their sort of concerns to us, really prominent, sort of a typical year of green spruce aphid. You sort of see this defoliation on the edge of a stand perhaps, but this has sort of gone all the way in. You can see the spruce affected there. Again, this is the one sort of looking into the distance. That's also between Cockermouth and Penrith. This is just west of Keswick. Again, really dramatic, sort of if you were seeing that for the first time. But as I said, if we go up a bit closer, I'm sure we'll be finding some sort of green tips to the green tip foliage in there. And this is kindly shared by Ian Jack from the Lowther Estates, who I think is on the call, so thank you. He sort of sent us an email with some lovely photos that I had to plug in where we can get a closer look. So again, you can see on this one, you've still got those green tips on the end of the tree. So we know that's doing okay, and that's going to recover over the next few years. But you can sort of see why people were so alarmed. So I've already given this away, sort of thinking, why have we had this boom year? Well, really mild winter. So it was the 5th warmest December on record. The winter just gone. You can sort of see that skewed towards the north, with Cumbria sort of just fitting within that. So mild winters mean you get less of a drop off of the population in winter than you would other years. But also thinking a warm and dry spring. So it's thought that these conditions perhaps exacerbate needle drop, especially the higher temperatures we've experienced in spring, and then obviously drought stress that might be experienced, that could also be exacerbating the stress on these spruce trees. So thinking about what you can do about it, because that's one of the questions we've been getting a lot. I point you again to Tree Alert. So Molly Davidson, who might be still hanging around on the call, has asked that we submit reports. So you might manage, a whole woodland, but you just need one tree alert report. Make sure you get photos of one tree so we can get sort of, wider aspect, but also you've got the symptoms up close, getting those sort of green shoots in there, so we can confirm it is green spruce aphid. And then include in your comments, you can then talk about symptoms in the wider forest. And this is to be really good for us to build evidence for what's going on this year, and sort of perhaps building a picture about where we're seeing it more in other parts of the country. Like I say, from the aerial analysis, it definitely seems like across the country, green spruce aphid and this defoliation seems more prevalent in the northwest, perhaps moving slightly into the northeast. And through the grapevine, I've also heard, obviously in Scotland and stuff as well, they've been picking up a lot of this defoliation. So having evidence for that, really useful for us. And then thinking ahead, sort of keep monitoring, and you can do a follow-up report if high levels of damage are still seen after flushing. So after you've got those new needles coming out, if you're still seeing high levels of damage or defoliation, then submitting a subsequent report next year. Yeah, and that's everything on green spruce aphid. So if anyone's got any questions, I'll quickly turn to the chat. See if anything's popped up. Thank you, Anna, for sharing Tree Alert. Yes, Nathaniel, go for it.
Hiya, Bronte. Just a quick one relating to secondary infestations on spruce that are infested by Elatobium. Have you got any sort of idea of what the potential of a secondary infestation of, say, Dendroctonus micans, or even potentially Stipographus in the future, might be on those infested by Elatobium?
Yeah, so Elatobium obviously reduces the tree vigor because you're losing that photosynthetic capacity with that needle drop. So you get a reduction in leader extension and also sort of growth of the timber. So I think there might be an association, and I see Molly has stuck her hand up, who is an entomologist with a keen interest in this. So I might let you take that question, Molly.
Yeah, absolutely. So to date, we haven't seen any link between if a tree with Elatobium is more likely to be infested by any kind of bark beetle. Narrowing down that direct link is really difficult to find out, but in terms of, as Bronte's already said, that's going to increase the stress of a tree, so that might then weaken its defences slightly. And we know that D. micans is a primary pest, so it will infest perfectly healthy trees otherwise. But it's certainly an interesting question and one that we've gotten quite a lot this year, so we might try and investigate it in the future.
Thanks, Molly. Any more? Any hands? Any questions? No, well, perfect. In which case, that is, stop presenting. That sort of brings us to the end of this Northwest Treehouse Group meeting. Thank you so much, firstly, to the speakers, Carrie, Anna, Mick. Thank you so much for taking the time to present to us. That was some really interesting stuff, and I think we're all going to have to go out now and stare at some plane trees and lime trees. Yeah, see what we can find and submit our tree alerts. I will say that we are planning the next Northwest Treehouse Group in-person event for this year, so I can say that is going ahead, which I'm very pleased to say. You can expect an email about that sort of into late summer, anticipating holding that in autumn as per usual. And I'll be sharing an email after this, sort of sharing links and things to all that stuff that the speakers have mentioned, so you can sort of follow up. But look, that's perfect. We've finished right on time. So thank you, everyone. And hopefully see you at the in-person event later this year.
Thanks, Bronte.
Thanks, Bronte.
and everyone.
It was really
Thank you.
enjoyable.
Thanks, everyone.
Bye.
Bye.
Yep,
Bye.
cheers.